Good morning. It's February 1st and this is your daily brief in science. Here's everything you need to know. Researchers from Saarland University in Germany have uncovered intriguing insights into the relationship between human ear movement and listening effort. Led by Andreas Schreuer, the study employed electromyography to objectively measure muscle activity, moving beyond subjective reporting methods. This research builds upon earlier findings from the year 2020, when Daniel Strauss's team discovered that auricular muscles activate in response to sounds coming from various directions. The current study indicates that the superior auricular muscles become more active during challenging listening tasks, suggesting an effort to enhance hearing ability. However, despite this activation, the effectiveness of these muscles in sound localization is limited due to their evolutionary decline over the past 25 million years. Historically, human ancestors could swivel their ears for improved sound detection, a trait still seen in modern cats and dogs. Today, humans have lost this ability, retaining only what researchers call a neural fossil. Interestingly, most participants in the study were unable to voluntarily move their ears, which indicates that the results were not influenced by this capability. In many animals, ear movement is essential for focusing attention and determining sound direction. The study sample was small, primarily consisting of younger individuals without hearing issues, which emphasizes the need for further research involving more diverse populations. The research team highlighted the importance of broader studies while acknowledging these findings as valuable insights into human auditory processing. Future research should also explore additional variables, such as eye movements and facial expressions, which may impact auricular muscle activity, according to researcher Yusuf Kakmak. An international research team has made significant strides in understanding the history of sheep domestication, revealing a timeline that spans over 12,000 years. The collaboration includes experts from Trinity College Dublin and LMU Munich, who analyzed 118 genetic samples from archaeological sheep bones discovered from Mongolia to Ireland. The findings indicate that the earliest evidence of sheep herding was located at Asikli Hoyuk in central Turkey, suggesting that domestication began over 11,000 years ago in the northern Fertile Crescent. Early herders are believed to have domesticated wild mouflon, which led to selective breeding practices that have shaped sheep populations globally. By around 8,000 years ago, European farmers began to selectively breed sheep for specific traits, particularly coat color. This marks the earliest known human influence on animal genetics, with research identifying genetic selection near the KIT gene, which is associated with white coat color. This suggests that early farmers preferred lighter-colored sheep for practical and aesthetic reasons. Dr. Kevin Daly highlighted the importance of sheep migration into Europe during the Bronze Age, which coincided with human migrations of the same period. Approximately 5,000 years ago, pastoralist migrations from the Eurasian steppes introduced significant genetic changes to both human and sheep populations in Europe, including the spread of Indo-European languages. Sheep have been closely tied to human societies for over 11,000 years, providing essential resources like meat, milk, and wool. This research underscores the mutual dependence between humans and sheep throughout history, illustrating their role in shaping civilization and agricultural practices. As human societies expanded, sheep populations became geographically distinct, but later blending occurred due to trade and migration, maintaining genetic continuity across regions. Ongoing genetic studies remain crucial for understanding the historical and contemporary significance of sheep breeding and their adaptation to human needs and environmental changes. Around 7,000 years ago, sheep genomes began moving west within the Fertile Crescent, paralleling human cultural expansion from Mesopotamia. Biomedical engineers at Duke University have unveiled an innovative AI-based platform called PepParClip. This groundbreaking technology is designed to create short proteins or peptides that can effectively bind to and degrade disease-causing proteins that have previously been considered undruggable. PepParClip consists of two main components: PepPR, which generates peptides through a generative algorithm trained on a vast library of natural protein sequences, and CLIP, which screens these peptides based on their amino acid sequences. The clip component is particularly notable as it draws inspiration from OpenAI's algorithm, allowing for the matching of peptides with target proteins without needing to know their 3D structures. This capability is crucial because over 80% of disease-causing proteins are disordered and tangled, making them difficult targets for traditional therapies. Peptides, being smaller than proteins, can bind to a variety of amino acid sequences, providing a potential solution for these complex proteins. The success of PepPar-Clip in targeting such challenging proteins opens new clinical possibilities for treating diseases that have long been difficult to address. In initial tests, the platform was able to design peptides targeting beta-catenin, a disordered protein associated with various cancers. Remarkably, 4 out of 6 peptides successfully bound to and degraded beta-catenin, effectively slowing cancer signaling. The research team also tested PepPar-Clip on a highly disordered protein linked to synovial sarcoma, achieving promising results in binding and degrading the target. In comparative tests, PepPar-Clip outperformed existing peptide generation platforms, such as RF diffusion, by producing better matching peptides more rapidly. Initial experiments showed that peptides designed by PepPar-Clip could bind to and inhibit the activity of the stable enzyme ultra-YD. Looking ahead, the research team aims to enhance the PepPar-Clip platform further and collaborate with medical professionals to develop therapies targeting unstable proteins involved in diseases like Alexander's disease and various cancers. These findings were published on January 22, 2025, in the journal Science Advances, emphasizing the potential of this technology to revolutionize treatment options. A recent study has uncovered a fascinating ability in fruit fly larvae. They can perceive both taste and texture when selecting their food, particularly decaying flesh. This research, published in the open access journal PLOS Biology on January 30, 2025, was conducted by Nikita Komarov and Simon Sprecher from the University of Fribourg in Switzerland. The study identified specialized neurons in the mouths of these larvae that are capable of detecting food texture. By selectively disabling taste neurons, the researchers found that the larvae lost their ability to sense food texture, leading them to consume food outside their preferred texture range. The experiments revealed a distinct preference for a specific texture of agarose, a sugary gel, indicating a Goldilocks preference for hardness that closely resembles days-old decaying fruit. These findings contribute to a growing body of evidence suggesting that neurons may have a multimodal capacity, responding to various sensory inputs beyond traditional taste signals. This challenges the conventional view that specific neurons are solely responsible for transmitting single taste signals, such as sweet or bitter. The study highlights the necessity of mechanical sensation for tasting texture, which is different from the chemical sensation required for flavors. Notably, the C6 neuron in the maggot's taste organ can sense both sugar and mechanical stimulation, showcasing its unique ability to integrate taste and texture information. Additionally, the research pointed out that the painless mechanoreceptor gene is essential for sensing food texture, emphasizing its significance in the gustatory profile. The authors stress the importance of food texture in overall food fitness and suggest that further investigations are needed to understand taste perception in mammals, including humans. This study expands our understanding of taste perception by identifying mechanoreceptors in fly peripheral taste organs that detect texture, shedding light on the complex interplay between taste and texture in food selection. In a groundbreaking discovery for marine biology, recent research has confirmed the first documented case of killer whale predation on a white shark in Australia. This significant finding sheds light on the complex interactions within marine ecosystems and the feeding behaviors of apex predators. The study, led by PhD candidate Isabella Reeves from Flinders University, identified distinctive bite wounds on the shark, indicating that killer whales specifically targeted the liver, a vital organ. This analysis not only confirmed the predation event, but also noted the presence of scavenging sharks at the site, suggesting a complex ecological interaction. These findings imply that killer whale predation on white sharks may be more common globally than previously understood, expanding the known range of their prey beyond seals. The bite patterns observed in this case were similar to those seen in killer whale attacks on white sharks in South Africa, emphasizing their selective feeding behavior. Dr. Alison Towner from Rhodes University highlighted that similar predation events in regions like South Africa and California have previously disrupted local shark populations, although direct observations remain rare. Associate Professor Adam Miller stressed the necessity of monitoring these interactions to grasp their ecological significance and the potential impacts on marine ecosystems. In October 2023, a 4.7-meter great white shark washed up near Cape Bridgewater, Victoria, and was confirmed as a victim of killer whale predation. 2 killer whales involved in this event, named Bent Tip and Ripple, were identified through a citizen science program tracking their sightings and behaviors. Witnesses reported observing these killer whales engaging in hunting behavior shortly before the shark carcass was discovered. The research, supported by multiple academic institutions, was published on January 27, 2025 in the journal Ecology and Evolution. The implications of killer whale predation are significant, as white sharks play a crucial role in regulating marine ecosystems, and their displacement could have far-reaching effects on ecological balance. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.